So today, we're going to unravel a medical mystery that's been unfolding for centuries. It's a story that takes us from something you can see with your own eyes, a devastating disease, all the way down to a single invisible typo in our genetic code. This is a journey of incredible observation and brilliant science, so let's dive right in.
Okay, so our story starts here, back in the 19th century. You have these physicians who are just trying to get a handle on this really perplexing condition that was affecting young boys. They saw kids who couldn't run, who struggled to even stand up, and who all did this very specific thing to get on their feet. Dr. William Gowers described it so perfectly back in 1879, it became the defining sign of the disease, a clue that would echo for decades.
Now, this investigation was a slow burn, for sure. You have Dr. Guillaume Duchenne, and at first he thinks the problem's gotta be in the brain. But he kept digging. By 1868, he literally invented something called a histological harpoon. I mean, think about that. To perform a very early, and I'm sure pretty brave, form of muscle biopsy. What he saw in that tissue sample changed everything, and he renamed the disease to focus on the muscle. That was a huge shift in thinking.
Right, so this brought up the huge question. Where's the problem? Is it in the wiring, the nervous system, or is it in the engine, the muscle itself? Pinpointing where the failure was happening was absolutely the essential next step.
And this right here is the moment everything changed. You had the legendary neurologist Jean-Martin Charcot look at the spinal cords of these boys, and what did he find? In his own words, absolutely negative. Nothing. The wiring was totally fine. That ruled out the nervous system and proved Duchenne's hypothesis was right all along. The investigation was now laser-focused on the muscle tissue itself, which, as you can see, was clearly degenerating. So the problem wasn't the signals being sent from the brain. It was the muscle fibers themselves that were failing to do their job. The muscle, that was the scene of the crime. And with that settled, the investigation could zoom way in, from the whole body down to the microscopic level. Okay, so now they knew where to look, but the next question was just huge. What inside the muscle cell was actually breaking down? Scientists had to go from looking at the tissue to searching for the one specific molecular part that was failing under pressure.
And eventually they found it. It's this whole collection of proteins called the dystrophin glycoprotein complex, or DGC for short. Now, the best way to think about this thing is like a molecular anchor, or maybe a shock absorber for the cell. Its whole job is to connect the cell's internal skeleton to the structure outside the cell, giving the muscle fiber all its strength and resilience when it moves.
And this gets us right to the heart of the matter. When any piece of this molecular anchor is broken or just isn't there, the muscle cell's membrane becomes super fragile. So the very things we take for granted, walking, standing up, those simple stresses are literally enough to tear the muscle fibers apart from the inside out. And here's what's so wild. This one complex holds the key to understanding a whole family of different muscular dystrophies. So if the main protein, dystrophin, is messed up, you get Duchenne. If the issue's with these other parts, called sarcoglycans, you get a different kind, limb-girdled MD. It's one molecular system, but a break in different parts leads to different but related diseases.
So once scientists figured out the mechanical problem, this broken anchor, right? They could trace it all the way back to its source, a typo in the genetic instruction manual. And you know what? Once you find a typo, well, you can start thinking about how to fix it. And that brings us into the modern age of therapy.
So how do you fix a genetic typo? Well, one of the cleverest strategies out there is something called exon skipping. Instead of trying to rewrite an entire gene, which is a massive job, this approach uses a tiny synthetic piece of molecular tape. It's not rewriting the whole instruction book. It's just covering up the one misspelled word so the cell can read the rest of the sentence correctly.
Okay, so here's how this incredible process actually works. You start with that typo in the gene. This little molecular patch, the AON, is designed to stick right onto that faulty section. So when the cell's machinery comes along to read the instructions, it gets to that part, sees the patch, and just hops right over it. It skips the typo. The end result is a protein that's maybe a little shorter than it's supposed to be, but the important part is it works. It can restore that anchor and protect the muscle.
And look, this isn't some sci-fi concept.
This is real medicine happening right now. There's an FDA-approved drug called eteplirsen that does exactly this. It targets a specific typo on what's called exon 51. And for roughly 14% of kids with Duchenne, this one drug, based on this one incredibly clever idea, can actually help.
So just to be clear, exon skipping is all about patching the original faulty instructions. It's a repair job. That's a little different from something like gene therapy, which is way more ambitious. Gene therapy is about delivering a whole brand new, correct copy of the instructions, usually with a harmless virus. Two different strategies, both aiming for the same goal: fix that broken anchor.
You know, when you take a step back and look at the whole story, the scientific journey here is just absolutely incredible. We've gone from simply observing a problem to directly intervening in our own genetic code. I mean, think about it. We went from a doctor in the 19th century just watching a child struggle to stand up, to a microscopic crime scene inside the muscle, then to pinpointing the single broken molecular anchor, and now all the way to designing therapies that can literally go in and tell our cells to ignore a genetic error. It's just a beautiful example of the scientific process in action.
And that just leaves you with this one powerful thought. The roadmap that scientists use to figure out muscular dystrophy, that can be used for other diseases too. As we get better and better at reading and even editing our own genes, the question really isn't if we can solve these devastating diseases anymore. It's which one are we going to unravel next?